Hello everyone, welcome to our presentation. Our topic is about introduction to membrane potential, action potential, and electrocardiography. Our team contains a panel of 10 of our dearest friends. We'll right away jump into our presentation.
Do you ever wonder how our cells communicate? Human cells communicate with the help of membrane potential. That is, by electric impulse, our cells coordinate with each other. The resting membrane potential, abbreviated RMP, is the potential difference between the inside and outside of the cell membrane. Usually, the membrane potential are the range of negative 70 millivolts to negative 40 millivolts. This resting potential is brought by permeability of the cell membrane of different kinds of ions that some ions can move across the cell membrane more easily than others. This is how the concentration of different ions is different inside and outside of the cell, creating creating a concentration gradient. And this concentration gradient provides a necessary potential energy to from the resting membrane potential. That means the membrane is polarized like a tiny little battery. Typically, the ions found in the most of the cells are positively charged and categorized, which include sodium and potassium, and negatively charged anions that includes chloride and organic anions. So now we clearly know that the diffusion of ions cause the exterior surface of cells membrane to get positive charge, while the interior surface accumulates negative charge, creating a electrochemical potential difference. As a result, stable resting membrane potential is established across the membrane where there is a dynamic equilibrium. The membrane is also selectively permeable. That is, it allows sodium ions more than potassium and chloride. The negative signs in the reading of RMP indicates that the inside of neurons of 70 millivolts lesser than the outside relative to each other. The inside of the cell has higher concentration of anions like chloride and proteins compared to the outside, which has more sodium and thus creating a gradient.
Now let us see about the ions involved in the membrane potential. Na plus has a much higher concentration outside of the cell, and the cell membrane is very impermeable to sodium ions. But K plus has a high concentration inside the cell due to the fact that the cell membrane is very permeable to K plus. And A minus is usually referred to large anions that are found completely inside of the cell and cannot penetrate the cell membrane. And the resting membrane potential varies according to types of cells. For example, skeletal muscle cells minus 95 millivolts, smooth muscle cells minus 50 millivolts, and etc. Yeah. And then the resting membrane potential, the maintenance of the resting membrane potential. RMP is produced and maintained by, firstly, the Donan effect. It describes that large, impermeable, negatively charged intracellular molecules attract positively charged ions and repels negative ones. And secondly, the membrane selectivity, which means the difference in permeabilities in the cell membrane towards different ions. That means some ions can enter the cell membrane easily, but some cannot. And finally, active transport. It is the mediated process of moving particles across a biological membrane against the concentration gradient. It comes under two mechanisms. Primary active transport involves energy, that is, it spends energy. This is how the innate ATPase pump functions. The secondary active transport involves an electrochemical gradient. This is not involved in maintaining RMP.
Action potential. Over time, the action potential can specifically change to transmit information. An action potential occur when the membrane potential of a specific cell rapidly rises and falls. This depolarization then causes adjacent location to similarly depolarize. Action potential are generated by special type of voltage gate ion channel embedded in a cell's plasma membrane. These channels are shut when the membrane potential is near the negative resting potential of the cell, but they rapidly begin to open if the membrane potential increases to a precisely defined threshold voltage, depolarizing the transmembrane potential. The course of action potential can be divided into five parts: rising phase, the peak phase, the falling phase, the undershoot phase, and the refractory period. Stimulation. A stimulus from sensory cell or another neuron causes the target cell to depolarize towards the threshold potential. If threshold of excitation is reached, all plus channel ion open and membrane depolarize. At peak potential, K plus channel open and K plus begin to leave the cell. At the same time, Na plus channel close. The membrane become depolarized as K plus ion continue to leave the cell. The hyperpolarized membrane is in a refractory period and cannot fire. K plus channel close and Na plus slash K plus transporters restore the resting potential. Okay. Resting potential. Neuron membrane is polarized with more negative ion inside. Depolarization. Stimulus trigger the voltage gated sodium channel to open. Depolarization is a process by which membrane potential becomes less negative, facilitating the generation of action potential. For this rapid change to take place within the interior of cell, several events must occur along the plasma membrane of the cell. So, sodium influx. Sodium ion rush into the neuron, reversing the membrane potential.
Now let's discuss how depolarization happens. Depolarization happens by stimulus activates voltage-gated sodium channels. Sodium ions rush into the neuron. Interior of the neuron becomes less negative. Membrane potential approaches zero, initiating an action potential. Repolarization occurs due to closure of voltage-gated Na plus channels and opening of voltage-gated K plus channels. K plus moves out of the cell into the interstitial fluid and polarity is reversed from positive to negative. Hyperpolarization phase of action potential occurs when voltage-gated K plus channels stay open longer than the time needed to reach resting membrane potential. During this time, the membrane potential is less than the resting membrane potential of minus 70 millivolt. Now, these are the phases of resting membrane potential. Graded potentials reach threshold. Third phase is depolarization. Fourth phase is repolarization. And fifth phase is hyperpolarization. And in the sixth and final phase being return to resting membrane potential.
Now let us see two effects governing action potential. Gibbs-Donnan membrane equilibrium, all or none law. The Gibbs-Donnan effect describes the unequal distribution of permeant charge ions on either side of semi-permeable membrane which occurs in the presence of impermeant charge ions. All or none law. If threshold reached, action potential propagates. If threshold not reached, not propagates. Same intensity of response to values greater than threshold. Nerve impulses are conducted by two different mechanisms. Continuous conduction, saltatory conduction. Continuous conduction occurs in unmyelinated action, sequential opening of voltage-gated sodium and potassium channels. Saltatory conduction occurs in unmyelinated actions, action potential propagated only at node of Ranvier. Myelinated region, as with limited numbers of voltage-gated sodium and potassium channel well insulated preventing ion movement. Nodes of Ranvier has large number of voltage gated potassium and sodium channels lack myelin insulation. Action potential disorder. Action potential disorder involve disruption in the generation or propagation of electric impulses within neurons. For example, multiple sclerosis, where the myelin sheath surrounding neurons is damaged, leading to impaired conduction of action potential. Epilepsy, characterized by abnormal and excessive neuronal firing, resulting in recurrent seizures. Parkinson's disease, too many excitatory stimuli causes the muscle to move without control.
Electrocardiogram. Before knowing about electrocardiogram, a prior knowledge about electric conduction systems of heart is essential. It has three main functions. 1. Impulse generation is done by the pacemaker cells in the sinus, that is, SA node and atrioventricular node, AV node. 2. Secondly, impulse conduction done by the cells of his bundle and Purkinje's fiber, which are thicker, elongated, and fast conducting. 3. Synchronizations of the atrials and ventricular work and between the pulmonary and systematic circulations is quite essential for the smooth functionings of the heart. Diagram. In this diagram, we can see the SA node, AV node, and left and right bundles fiber. We want to clarify first itself that electrocardiogram is the line graph we obtain and electrocardiograph is the machines we use to measure the heart impulse, keeping that in mind. Now let us see about ECG. It is a diagnostic test that a healthcare professional uses to measure the electrical activity of a person's heart. It records the heart electrical changes in a form. The animations below shows the normal cardiac conduction and its corresponding wave positions in the ECG graph.
The physics behind this ECG is rather very familiar. Can you guess what a CCG actually measures? It measures distinct potential difference on the surface of the body. But from where that potential difference comes from? We know that the cardiac muscle cell depolarize as they contract. This moving depolarization corresponds to a moving electric dipole, which produces an electric field on the body surface. This varying electric field can be detected from the surface of the body. In the heart, for each heartbeat, depolarization initiates in SA node and spreads throughout. Repolarization occurs after a significant delay. As depolarization spreads, the heart's total dipole moment at any instant in time is the vector sum of all the dipole moment of the individual parts. Because the cells are not at all activated simultaneously, but are activated in an XYT timed sequence in order for the heart to function. This total dipole moment changes both the magnitude and direction throughout a single heartbeat. For each measured potential difference, the two electrodes are roughly equidistant from the heart and on opposite side of it. Consequently, each potential difference is proportional to the component of heart's electric dipole moment along the axis on which it measured. Its time depends reveals how the component changes due to the movement of the dipole moment. The entire dipole moment can be reconstructed from the measured set of potential difference. Do you realize the heart's electric field is much dominant than other cells? But why? This is due to the coordinated wave of depolarization combined with the delay before the repolarization, which is longer in cardiac muscle cells than in other cell types, causes the heart to be the dominant source of the electric field on the body surface.
All simultaneous action potentials merge into the P wave, the atria, QRST ventricles. The cycle diagram depicts the heartbeat of the continuously repeating cardiac cycle, namely ventricular diastole followed by ventricular systole, coordinating with arterial systole followed by the arterial diastole. The key also correlates to the key electrocardiogram trackings. The T wave indicates ventricular diastole, the P wave arterial diastole, the QRS spikes complex ventricular systole. The atria. ECG shows small arterial deflection, reflecting a small muscle mass. Depolarization P wave, rounded P slow conduction. The ventricles. Large deflection in ECG as the chamber muscles are thick. Depolarization QRS. Steep peaks, fast conduction and repolarization result in T wave. Left ventricle, main contributor to QRS. Electrocardiogram. P wave. The P wave is the first upward deflection seen on ECG tracking. Significance: it represents arterial depolarization, which is electrical activation of the atria as they contract to push blood into the ventricles. Duration: normally the duration of the P wave is between 0.06 to 0.11 seconds. QRS complex. The QRS complex is a series of three waves Q, R, and S. Significance: it represents ventricular depolarization, which is the electrical activity of ventricles as they contract to pump blood out of the heart.
Normally, the duration of the QRS complex is less than 0.12 seconds.
Okay. ST segment. QRS terminates at J point, the point where the steep ends of QRS suddenly flattens and mark the beginning of the ST segment. It is the time during which all the regions of the ventricles are completely depolarized, lasts for 0.09 seconds. T waves and QT times. The T waves represent ventricle repolarization. The wave starts with slope but has a steep return to baseline. The QT times reflects the duration of the systoles and it's measured from the onset of the earliest Q waves to the end of the T waves. That is the point where the tangent to the steepest downslope of the T waves across the baseline. The action potentials are shortened when the heart beats faster so that the QT times is long in bradycardia and short in tachycardia. Eingovan's triangles, recording method for hot electrical activity. Choose the point that have the greatest voltage difference. U. Eingovan's triangles with the three leads. Record U between the left arm positive and the right arm negative. Record U between right arm negative and left leg positive. Record U between left arm negative and left leg positive.
Now, let us see about the coping and treatment strategies for abnormal ECG. The disrupting the sequence of electrical activation or changing the heart's structure changes the magnitude, direction, or time dependence of the heart's dipole moment vector significantly, giving the ECG its diagnostic power. And we normally use defibrillator, which is a treatment for life-threatening cardiac arrhythmias, specifically ventricular fibrillation and non-perfusing ventricular tachycardia. We deliver a dose of electric current and restart the heart's nodal systems. And next, artificial pacemaker. Let us look a video about it, where the pacemaker regulates abnormal impulses. Thank you all.